Okay, so thank you all for having me here today. I'm excited to talk to you about my research. And I was asked to begin this talk with a discussion of sort of what got me interested in neuroscience and talk a little bit about my educational path before sort of then diving into some of the scientific work that I've been engaged in here at EKU. What got me interested in neuroscience? So as mentioned, I attended college in Louisiana at this kind of very small liberal arts college named Centenary College of Louisiana. And I was definitely one of those students at Centenary who struggled quite a bit to choose a major. So I think I didn't choose until close to my junior year. I started off as a political science major and was thinking, maybe one day I'd go to law school. And so I took some classes in political science, but after a while, I kind of began to lose interest. And then I started thinking, maybe I want to do computer science. I'm definitely, my whole life, I've been a big computer nerd. I mean, big computer nerd. This is a picture of me, like 16 years old in high school doing a science fair project to benchmark computer hardware. And so I thought maybe I'd go that route. But we had a pretty small computer science program at my school that was really kind of just under development. So then I thought maybe I would go into kind of a more established department and do something sort of related. Maybe I'd become a mathematician. So I took a variety of courses in math, but by the time I got up to about calculus 3, I started questioning, do I really want to make a full career out of mathematics? And all the while, I was taking courses in psychology. And really, the thing that intrigued me about psychology was trying to understand the factors that drive or kind of what we become, right? Our thoughts, our emotions, our behaviors. And I really found that, those ideas to be interesting. But as I took courses in psychology, one of the things that I realized I didn't like about the field was that it involves sort of an outward-in approach. So you're sort of studying the behavior of animals to infer what is happening in the brain. And there was just part of me that wanted to somehow, at sort of a more mechanical level, to really understand sort of the nuts and the bolts of the brain and really kind of dive in deep to understand how the brain is functioning. And so then that kind of got me thinking about biology. And biology is a topic that I've long had at least some interest in because I've had a lot of family influence related to biology. So I've got a grandmother who is an expert in mollusks. My dad studies hormone responses to exercise. My mother was a nurse. I had an older brother, who's a physician. And so you can imagine around the dinner table, the concepts related to biology would come as I was growing up.
It's you not the move tonight. The only thing I like about it is like the color coding they're doing.
Yeah. So maybe we could that interest in biology.
It's small too. Yeah, like that's what I'm saying. It's smaller. ATO is literally packed full. Like I feel like I can't even breathe.
Thank you. Okay, so I talked to a college advisor who then kind of helped me to learn about this field, neuroscience. And as I learned more about it, I realized this was exactly what I wanted to do. So this is a field that's focused on understanding the central nervous system, this organ system that controls every other organ in the body. And that is the basis for sort of who we are, right? It controls our thoughts, our imaginations, our motivations, our behaviors. And what's really cool about neuroscience is it's a really interdisciplinary field. So it draws upon approaches from biology to psychology. Computational analyses are really important for the field. Math and physics and medicine, all these sort of approaches in these various fields are utilized to understand the function of the nervous system. So I started taking courses in neuroscience and realized that, that I loved it, but then the question is, what do you do with that degree? So my first thought was, okay, maybe I'm going to be a physician. So I studied really hard for the MCAT and quickly took it because I was running out of time. I didn't know a neurologist, but I had a family practice physician, and so I shadowed him. And I kind of thought the experience would be sort of like one of these TV shows, you're kind of really diving in deep to understand, mysterious illnesses. What I saw instead was sort of a lot of colds, right? And I was surprised that I didn't find it to be I wasn't quite as captivated by it as what I was hoping. And so I began to think about what might be better options for me. And so I engaged in my first research experience. So there was a mentor, and my undergraduate mentor, his name was Greg Butcher, and I did some work with him on trying to understand the molecular basis of circadian rhythm, so how our bodies can keep time. And I had the opportunity to go to an international meeting. So this SFN meeting usually has about 36,000 attendees. So it was kind of fascinating to see all these neuroscientists across the world that kind of, were interested in understanding how the brain works. So I decided to go to graduate school. I ended up going to Vanderbilt University, where I got to do rotations in a variety of neuroscience-related labs. And I ultimately joined a lab of this person, Dr. Bruce Carter, and began training to understand how to study cell signaling cascades that affect neuronal survival. I also knew I would be really interested in teaching. And so while I was in grad school, I engaged in a certificate program to learn how to teach at the college level. And then when I finished, I did something called a postdoctoral fellowship. For any of you who aren't familiar, that is sort of after you get your PhD, an experience where you can continue to do some additional training in a lab before eventually going on to a faculty position. And so I actually did some work with a clinician. So this was Dr. Laura Dugan. She's the chair of Alzheimer's and geriatric medicine. And so I learned, I got some training in the aging brain and how inflammation might contribute to the development of disorders such as Alzheimer's disease. Okay, so that's a little bit of my educational path and why I'm interested in neuroscience. In terms of what a day is like in the lab, I think the best way to share that is just to kind of describe some of the research that I've been doing. So I'm going to talk in particular about two projects today. The second one I'm probably going to, for the sake of time, just kind of skim over, and we'll dive into the first one in a little bit more detail. So this first project relates to a protein termed the P75 neurotrophin receptor, and it understanding the way that it signals and how that might be important for a disorder such as Parkinson's disease. And all of the work I'll be talking about today was done here on campus with valuable contributions from a variety of students here at EKU that I've really gotten to enjoy working with. Okay, so for some background. So P75 is a transmembrane protein. So it hangs out in the membrane of cells. And it's a protein that can become activated and start signaling with other molecules if it gets activated by these proteins called neurotrophins. So neurotrophins are these molecules here. We have nerve growth factor, brain-derived neurotrophic factor, neurotrophin 3, and neurotrophin 4. And if any of these guys bind to P75, and that'll cause it to initiate some signaling inside the cell and make different things happen. And what exactly P75 can do can really vary depending on the cell type. So sometimes it can regulate how neurons become myelinated, how they get sort of wrapped with fat in a way that allows them to conduct electrical impulses. P75 can affect synaptic plasticity, sort of the wiring of different neurons in the brain. But among its various functions, I'm particularly interested in its ability to do this. So there's been a variety of studies showing that in certain contexts, if P75 gets activated, it initiates a cell cascade that causes neurons to die. And I want to understand how that happens and how that might be important for understanding certain neurodegenerative diseases. Now, despite the fact that this receptor has been known about for a good 20 years, how it signals is still fairly poorly understood. But there is sort of one signaling mechanism used by this receptor that's been well established from work by a variety of research groups. And so this is sort of our traditional model of P75 signaling. So it's known that if any of those neurotrophins, like NGF or BDNF, NT3, NT4, if they bind to P75, then this somehow causes this receptor to become cleaved into some fragments. So there's this enzyme named TACE that will cleave the extracellular domain of P75. And then there's this other protein complex called the gamma secretase complex that'll cleave P75 in this transmembrane region here. And this is important because after this second cleavage step, this releases this intracellular domain of the receptor. And there's all sorts of different molecules that can bind to this intracellular domain and then go on to do other things, right? And so it's thought that release of this intracellular domain might enable this piece to then signal with some proteins to cause death in the cytosol, or maybe it could even go to the nucleus and contribute to regulation of gene transcription in a way that would ultimately lead to the cells dying. Exactly what happens downstream is sort of a matter of controversy and still a hot topic of exploration. But this upstream step, just the fact that it all starts with the receptor getting cut into pieces, is something that's been very well established. Okay, so where this might be important is in the context of various pathological conditions. So it's been shown that, if someone has seizures, so there's lots of neurons firing action potentials in sync that can cause neurons to die. And P75 has a really important role in that. If you have various injuries, like if a nerve gets severed, or that's called an axotomy, or if you have a spinal cord crush, again, P75 is contributing to the death of neurons in response to those types of injuries. P75 has also been shown to be upregulated, so levels of it are increased in certain models of diseases, such as models of Alzheimer's disease or amyotrophic lateral sclerosis. And so a question that I've had for a long time is, these are all very, very different types of pathological conditions. The reason a cell is dying in response to a seizure, totally different than the reason a cell is dying in response to, let's say, amyotrophic lateral sclerosis. So how is it that this one protein gets signals in response to such a wide variety of pathological conditions? And so, I first began to investigate this question as a graduate student. And with my mentor, we hypothesized there must be some very general response to stress that cells have, such as inflammation or oxidative stress, that leads to activation of this receptor. So for any of you who aren't familiar with oxidative stress is this condition in which you have an overabundance of these really reactive molecules called reactive oxygen species. So reactive oxygen species are usually prevalent in all types of cells, but usually they become detoxified by antioxidant molecules. But in a state of oxidative stress, you have an imbalance. So for some reason, you have too many reactive oxygen species compared to the antioxidants, and thus these reactive molecules can modify proteins, it can cause damage to DNA, that can impact lipids, and ultimately lead to the demise of the cell. Okay, so I'm not going to go into my graduate work, but just as to show you guys a model, one thing that I discovered with work in my graduate lab was indeed, if there's a condition that involves an elevation of reactive oxygen species, this does activate P75 and cause P75 signaling. And this discovery was made using sympathetic neurons, which are these neurons that are out in the body, and they are responsible for our responses to stress. And so we found that in sympathetic neurons, if you subject them to oxidative stress, that this causes P75 to get cleaved and sets off this whole signaling cascade. But what was really interesting is when we were performing these studies, we realized that this is somehow happening without that step where a neurotrophin has to first bind to the receptor. So neurotrophins weren't required for this activation of the receptor. So this work kind of revealed a new mechanism of P75 activation, what we call a ligand-independent mechanism of P75 activation, in which you don't have to get something binding to the extracellular domain first in order for the receptor to start signaling. And so this was kind of a pretty neat discovery, but from that graduate work, all of that work was done in sympathetic neurons that are out here in the body. And the way that receptors signal can be very different in different regions of the body and in different types of cells. So we had this remaining question, which is, does this novel mechanism of P75 activation, does it have any role in the brain? And could it be important for regulating neurodegeneration associated with various disorders that affect the brain? And so in kind of pondering that, the first disorder that came to mind and sort of thinking about a neurodegenerative disorder that involves oxidative stress is Parkinson's disease. So Parkinson's disease is a disorder that involves a progressive loss of these neurons in a region of the brain termed the substantia nigra. And these neurons communicate using the neurotransmitter dopamine. And so over time, as these neurons progressively die, you get less release of dopamine from these neurons onto this other region of the brain called the striatum. And that leads to a variety of motor deficits. So individuals have trouble with posture and with initiating movement, and it can eventually lead to some cognitive deficits as well. Now, P75 had been reported before to be expressed in these dopaminergic neurons, but how it signals there and what its function is in those cells hadn't been explored. And so we thought that would be a pretty neat thing to try to figure out. And so we hypothesized that maybe this novel mechanism of P75 activation in which oxidative stress causes the receptor to get cleaved in a ligand-independent manner, maybe that happens here. And maybe that could be a way that this receptor contributes to cell death associated with Parkinson's disease. Okay, so to test this, we needed some model, some way to, to sort of emulate Parkinson's disease. And we chose to do this using cell culture. So we have these pretty neat cells. They're called Lund human mesencephalic cells. And so they look a lot like just a regular cell line. They're these little cells and they divide very rapidly. You can get lots of them in a dish, and they're great for fast work with undergraduate students. But if you treat them with certain factors, then they differentiate. They start to grow out these beautiful axons and dendrites. They network to each other. They become electrically active. And so they become neurons. And importantly, they become dopaminergic neurons. So here you can see some staining for an enzyme called tyrosine hydroxylase. This is an enzyme that's involved in production of dopamine. So we can see that these cells have the machinery to make dopamine. And so the first thing we did is ask, do they have P75? And so we did some staining for P75. And we can see, indeed, P75 is there, and it's there in the cell bodies. It's also there throughout the neurites, throughout these axons and dendrites that neurons use to communicate. So we have a system where we've got mature neurons, they make dopamine, they have P75. So this is a good way to figure out how does P75 signal in dopaminergic cells. We needed some way to cause an injury that sort of models Parkinson's disease, right? And so for that, we chose to treat ourselves with this drug called 6-hydroxydopamine, or 6-OHDA. This is a drug that causes oxidative stress. So you can see if we put increasing amounts of this drug on, those neurites start to fragment and the cells start to die. And so if we look at, some quantification of this, we can see that with higher amounts of this drug, we see more and more death, right? And so this is our model for causing neurite degeneration in a dish of cells and then interrogating signaling mechanisms associated with that. Okay. So once we had sort of our model established, our first question, does oxidative stress cause P75 to become cleaved in these cells? And so to investigate this, we did what's called a Western blot. For anyone who's not familiar with the Western blot, it's a technique where what you ultimately get is sort of a membrane with these little bands on it. And the bigger a band is, the more protein that means you have there. And all of your proteins are separated by size. So based on where you see that band, that can tell you what the size of that protein is. So if P75 has not been cleaved into fragments and it's just nice in its full-length form, what we should see is a big band that's close to 75 kilodaltons with no little other bands at other sizes, right? And so in this lane, what we're looking at are healthy cells. So they've been treated with a vehicle solution, which is just like saline. And they're nice and healthy, and so we just have lots of intact P75, right? But if we treat the cells with our 6-OHDA, our drug that causes oxidative stress, now we see that this full-length version of the receptor is reduced. And concurrently, we're seeing these fragments show up at smaller sizes, which represent these pieces of P75 that remain after it gets cleaved. Right. And then this is a quantification of this. So we can see that this, after that first cleavage step, this remaining piece here is called the C-terminal fragment, and it's represented by this band here. We can see that levels of that are significantly increased in response to oxidative stress. This piece, this intracellular domain that's produced from the second cleavage step, it can be seen here, and its levels are significantly increased in response to oxidative stress. And then this band, which represents the full-length receptor here, we can see that levels are decreased. And so that's a significant decrease there in response to oxidative stress. Now, a good common approach in science is it's often good to verify a result using more than one method. So we also chose to kind of evaluate, ask the same question in using a different approach. So here what we did is we stained our cells with an antibody that'll only recognize P75 on the surface of the cell. And we did some confocal microscopy. And so what we can see here is in healthy cells, there's lots of, you can see that extracellular domain of P75 is hanging out there outside the surface of the cell. But in response to oxidative stress, the amount of cell surface exposed, the extracellular domain of P75 is reduced. And so this is indicative of that extracellular piece being cut off. And so it's no longer there to be detected by our antibody. And this is a quantification of that here. We can see this decrease in cell surface, P75 in response to 6-hydroxydopamine. Okay, so all of this is to say that P75 gets cleaved in dopaminergic neurons in response to oxidative stress. So our next question is, well, how is this happening? Is this more, is this fitting more with the traditional model where maybe, we have oxidative stress that somehow makes the cells produce neurotrophins, and then the neurotrophins bind to the receptor, and then this receptor gets cleaved? Or is this occurring through our newly discovered ligand-independent mechanism? Maybe oxidative stress somehow more directly activates the enzymes that cleave P75, and we get this cleavage happening without neurotrophins binding to the receptor first. So to test this, we decided to treat ourselves with this antibody that had been previously shown in other studies to bind to this extracellular domain of the receptor and block any neurotrophins from sticking to it, right? And so the idea here is if neurotrophins have to bind to P75 in order for P75 to become activated, well, this antibody is in the way, right? So with oxidative stress, well, if neurotrophins have to bind, it can't get there. And so that should keep this receptor from being cleaved, right? But if neurotrophins don't have to bind to P75 in order for the cleavage to happen, then we should see that cleavage happening anyway, right? So oxidative stress would occur and boom, we would still get cleavage of the receptor. Okay, so here's the data from these experiments. So we can see, our first two lanes are similar to the last experiment that we just described. We can see with healthy cells, we have lots of full-length P75, and that full-length P75 is reduced, and these fragments show up if we have treated the cells with 6-hydroxydopamine and caused oxidative stress. But if we first pre-treat with this antibody that blocks neurotrophins from binding to P75, we still see cleavage, right? We still see less of the full-length receptor, and we still see these fragments here. And so this indicates that neurotrophins don't have to bind to the receptor in order for that cleavage of the receptor to occur. And thus that this is happening via a ligand-independent signaling mechanism. Okay, we next asked, are the enzymes that are chopping this receptor, TACE and gamma secretase, those are enzymes that have been shown to cleave the receptor in response to neurotrophins binding. Is it the same enzymes that are doing that in this case, when we have ligand-independent activation of P75? So is it TACE that's doing this, cleaving this extracellular domain, and gamma secretase that's cleaving this transmembrane region? And I'm going to kind of quickly go over this slide due to the sake of time. So I'll just say that what we did is we used some drugs that block either TACE or gamma secretase, and we found that those drugs did block cleavage of the receptor. So we confirmed it is the same enzymes cleaving P75 in this case, as has been shown with more traditional context related to P75 cleavage. Okay, so then our next question was, well, how is this happening, right? So how does oxidative stress somehow lead to cleavage of the receptor if a ligand isn't necessary? And after doing some literature review, we keyed in on this one particular protein. It's got a long name, it's C-Jun N-terminal kinase, but it's often abbreviated J&K or junk, is what a lot of scientists call it. And so J&K is a protein that's been well known to become activated in response to oxidative stress in a lot of different contexts. And it's a very important signaling protein. It's involved in a variety of different signaling cascades. And so we thought, what if oxidative stress may stimulate junk activity And then junk, through various signaling mechanisms, somehow promotes activation of these enzymes that cleave the receptor. And so we tested that out. So we used a J&K inhibitor called SP600125. And so what we can see here, we did a Western blot assay. And again, if we've treated our cells with a drug that causes oxidative stress, we have less full-length receptor, and we have these fragments here. But if we treat with this J&K inhibitor, now the amount of cleavage of P75 is reduced. We have more of the full-length receptor and less of these fragments. And so this indicates that J&K is required for activation of P75 in response to oxidative stress. And something that was kind of cool is while we were doing these studies, as the students were looking at our cells in culture, what they noticed is, usually when you treat these cells with 6-hydroxydopamine, it kills them, right? They're going through oxidative stress, so they ultimately die. Well, they noticed that when they treated with this inhibitor, the cells looked a lot healthier. And so we decided to quantify that. So here you can see we've stained our cells with just a cytoskeletal, we're staining for a cytoskeletal protein here. We have healthy cells here. If we treat them with 6-hydroxydopamine, we see all these little dots. That's the neurite starting to fall apart on the cells. And we can see that the cultures look a lot healthier if we've given them this J&K inhibitor. And we quantify this, so we can actually zoom in on these little blue nuclei that we've stained for. And by looking at the size of those nuclei, we can evaluate whether they're healthy cells or dying cells. So what we can see is when we treat with a drug that causes oxidative stress, a lot of them die. But if we pre-treated with this inhibitor that blocks J&K signaling, a significant proportion of them are protected. And so, so to conclude here, wrap up this. We discovered that P75 is expressed in dopaminergic cells, that it becomes, and what we think happens is oxidative stress promotes junk signaling, and junk leads to cleavage of the receptor, first by TACE and then by gamma secretase, thus freeing the intracellular domain to ultimately promote death of the dopaminergic cells. Okay, so where do we want to go from here? So we're currently, so kind of a common theme in science is often if you discover something in cell culture, it's then valuable to confirm whether that phenomenon occurs in living organisms, right? And so we're now moving to some mouse models. And so to model Parkinson's disease in mice, we're doing some live animal surgery. So we're anesthetizing the mice, and then we're actually doing brain injections. So we're injecting 6-hydroxydopamine into the striatum of the brain. And so this is what this looks like. So if we look at the uninjected side of the brain, so this is the substantia nigra on the uninjected side, and in black, you're seeing all the dopaminergic neurons. And in response to our drug, you can see fewer of those dopaminergic neurons on the right side. Right. And then we're using two different drug-based models of Parkinson's disease. So there's another drug called rotenone where we kind of see a similar thing. On the injected side, we see fewer of these dopaminergic neurons. And so what we're currently doing is we're performing this procedure with normal mice and mice that lack a proper gene for P75. So these mice can't make P75. And so the question is, if they don't have P75, will these cells still die in response to the drugs that cause oxidative stress? And so that's some ongoing work. We also recently discovered a pretty cool phenotype in our P75 knockout mice. So we were staining the striatum. Remember, that's the region that those dopaminergic neurons project to. And we realized that even if we haven't done any surgeries, even if we're just looking at sort of the mice in an uninjured state, the P75 knockout mice have this dramatic reduction in the amount of dopaminergic neurites that are connected to the striatum than compared to a normal mouse. And so this actually suggests that even in the absence of a pathological condition, that perhaps this receptor actually has a role during neurodevelopment in either controlling how the dopaminergic neurons survive or maybe how those axons grow out to the striatum. And so we've got a cool new project that we're currently engaging in to understand what role P75 might have in the maintenance of these dopaminergic axons during early neurodevelopment. Okay, so due to time constraints, I'm not going to go through this second project in great detail. I'm just going to kind of give you guys a summary. But we've also been working on a second project that is sort of shifting gears a little bit away from P75, and it relates to sort of development of a new tool that investigators can use to perform studies related to neurodegeneration. And so what this tool relates to is it's a tool to help scientists perform studies to measure neurite degeneration. So with these different disorders like Alzheimer's disease or Parkinson's disease, a common thing that happens is before the cell dies, these neurites that neurons use to communicate, these axons, for example, they develop these little swellings, and then they start to fragment. And this happens first, and then later on, eventually the cell bodies die, right? And so neurite degeneration is an early stage event associated with a whole variety of neurodegenerative disorders. And so we need studies to understand what's causing these early stage events associated with neurodegeneration. But there's a problem. So if, let's say, an investigator has cultured cells and they're treating with drugs and they're doing various things that they think causes neurite degeneration, and then they want to measure that and say how much neurite degeneration has happened, there's this common technique that's used, but it has some limitations. The way that this technique works is you take a picture of your cells. So here we have some cells, and some of the neurites are degenerating, some of them look good. And using open source software called ImageJ, you binarize that image. So you make it all black cells on a white background. You want this image to have only neurites. So you digitally remove these cell bodies from the image. And then there's an algorithm that will detect particles of certain size and circularity. By modifying those parameters, you can get this software to detect the little fragments. And then you calculate what's called a degeneration index. So it's a measure of the area of the fragments divided by the total neurite area, right? And so another way to say it is sort of like out of all the neurites in the image, what proportion of them are fragments, right? Okay, so we in our lab wanted to do some studies related to neurite degeneration. And so we tried to use this very widely cited technique. And it sort of works, right? So here you can see if we treat with increasing amounts of 6-hydroxydopamine, and so we get increasing amounts of degeneration. You know, when we perform this method, we see increasing amounts of neurite degeneration detected by this measurement method, right? But while we were doing this, we noticed a few key issues. We noticed that sometimes there's this thing called artificial fragmentation that happens. Sometimes you'll have a picture where these neurites look perfectly healthy, but when you binarize the image, now they look fragmented. And that's going to create a lot of error, right? B, there's this issue where sometimes it's not detecting all the fragments. You know, so we see little red spots, that's the fragments that are detected. But with this method, look at all these fragments that we're missing, right? So the sensitivity of this method has some issues. That's very good. And then C, this method is tedious, right? So a lot of scientists use this method on what are called explant cultures. An explant culture is when you take some tissue and you plop it into a dish and let those neurites just grow out like this. And so with an image like this, if you want to make this image only have neurites, you only have to digitally remove just this one spot where the cell bodies are right here, right? But let's say we use what are called dissociated cultures. These are real commonly used cell cultures where you break up the cells into little individual cells and you let them form networks with each other and communicate with each other, right? Well, if you want this to be an image that only has neurites in it, now you have to manually circle and remove each cell body. You can imagine my students don't like doing this, right? And you can imagine that if it's a big image, you know, and if you have 200 of these images, it can take a long time. So we set out to try to automate this whole process. And so we developed a variety of improvements. We figured out a way to, so we decided to use cells that had been stained to get better contrast for our images. We developed a way to, in an automated fashion, remove the cell bodies. And the way that this basically works is we have staining of the nuclei of the cells here, staining of the cytoskeleton here, We binarize the image of the nuclei, and then
Hello.
we implement some operations to dilate those nuclei to make them bigger to encompass the size of the cell body. We subtract this image from the image of the neurons, and then we binarize that. And what we get is an image where all the cell bodies have been automatically removed. We have just the neurites there. Then we do that particle analysis operation, but we implemented a variety of enhancements to make it more sensitive and to remove not like cell debris that could confound the assay. And we also just put in a variety of other improvements to make it very seamless and user-friendly. And for the sake of time, what I'm just going to show is I'm going to skip to this data here. So what we did is we had some students in a blinded manner. They looked at a bunch of images of cells that were either healthy or degenerating, and we had them score subjectively on a scale of 0 to 1, how degenerated do these neurites look, right? And then we use the traditional method to actually measure the degeneration. We can see that the subjective measurements by blinded students correlated fairly well with sort of the measurements taken by this traditional method. However, if we used our new macro, which by the way is called ANDI, Automated Neurite Degeneration Index. So ANDI was able to give us scores that much more closely approximated the amount of neurite degeneration that blinded investigators reported seeing. And so this is sort of evidence that ANDI is producing measurements that more closely match what we can see with our own eyes when we're looking at these images. And of course, the biggest advantage to ANDI is the reduction in time. So it takes substantially less time to perform these analyses using this new program. Okay, I'm going to skip this. We kind of provided proof of principle of using this assay in an experiment. And we found that J and K, if you block it, it does protect the neurites from degeneration. And Andy was able to help make that revelation. And so I'll wrap up. So in conclusion, we developed an automated method for measuring neurite degeneration it's more accurate, more efficient. It can be used on dissociated cultures in a fast way. And because it's all automated, the great thing about it is you don't have to be an expert in image J. It's very easy to use. So for scientists that are just getting interested in this area and want to study neurite degeneration, it's very easy for them to apply this technique. And we just actually had a manuscript, I think it was last week, we received news that it was accepted. So the macro, the ANDI will be available in this manuscript. It's all open source, and so any investigators can use it. All right, so what's a day like in my lab? It can really vary, right? Sometimes I'm doing bench work with cells. Sometimes I'm doing a surgery on a mouse. Sometimes I'm writing code and developing software. Other times I'm working on grants, a lot of grants, making manuscripts, I'm purchasing supplies, and training students. So it can vary, but I think that's part of the fun of doing neuroscience research is that you can mix it up and take a variety of approaches, and it's not always sort of doing the same thing. All right, I'll stop there. I'd like to thank all the students that have contributed to all of the work that you guys have seen here, as well as funding sources. So a lot of this work has been funded by Kentucky Embray, formerly known as Cabrin, the College of Science here at EKU, the Department of Biological Sciences, and the Battelle EKU Science Scholars Program have all also supported this work. And with that, I will take any questions.
Thank you, Dr. Kraemer. So we'll open it up to in-person questions. So we'll get some time for folks on Zoom to answer their questions. So any questions in here? I have a question, actually.
Sure.
So we talked about the 6, I might be saying around the 6-ODHA.
6-hydroxydopamine.
Okay, yeah, OHD. Okay. So that drug, you mentioned that it sort of causes stress that degrades the neurite connection to fragments them. So it just makes me wonder, what is the purpose of that drug if if that negative side effect is so prevalent, like what is that even used for?
Oh, good question. So 6-hydroxydopamine is not a drug that's used for therapeutic purposes. So it was more specific. So it's actually so even though it's, when we buy it, it's typically synthesized in sort of in a lab, it is something that can occur naturally during oxidative stress. So 6-hydroxydopamine, so it's basically dopamine with a hydroxyl group on the 6th carbon. And so it's something that can naturally be produced, but it is used by scientists more for this type of reason, just to model a disease-like state as opposed to, having some benefit therapeutically.
Okay.
Yeah, great question.
Thank you.
Sure. Yes.
Brian, you mentioned this point a couple of times. P75 has been known for over 20 years, but it's been kind of pushed aside. Maybe it's a sociology of science, but why is that? It seems to have some pretty key implications here for Parkinson's, for example.
Yeah, good.
Why has the focus in the field not been on P75 for so long?
Yeah, great question. So it's a very promiscuous receptor. So there's been quite a bit of research on P75, but one of the challenges is it's a receptor that has 4 different ligands, plus all I didn't mention, but all 4 of those ligands can be modified in various ways, and that affects its affinity for the receptor versus other receptors. And then there's probably 25 to 30 different cytosolic signaling partners that have been discovered. And so the issue has been, one group will say, oh, this is the real key player. This is a new cytosolic signaling partner for P75, and that's how this receptor is signaling. And that's really important in the hippocampus. And then another research group will say, no, it's this protein, that's the key player, but we did our studies in the cerebellum, right? And so there's been a lot of work, but folks have gotten different results, and they've often started with different cell types. And so it's made it difficult to sort of put all this information together in a way where you can sort of reach a consensus about how this receptor is signaling. But that cleavage of the receptor is sort of the one thing where, in a variety of different cell types from a variety of different groups, that's been recognized to sort of serve as a starting point for this receptor initiating its signaling. Yeah, good question. Yes.
Is the cleavage business, is that known in any other kind of receptors? Is that sort of a general thing, or is that?
Yeah.
P75?
Yeah, very good question. So it is indeed something that happens with a variety of other transmembrane proteins as well. So probably the most famous example is that there's a protein called the amyloid precursor protein. And amyloid precursor protein gets cleaved by TACE and then by gamma secretase. And those fragments, there's actually another enzyme that can cleave it as well. And those fragments, one particular fragment is called the amyloid beta fragment, and it can go bind with other amyloid beta fragments and create this big glob that's called an amyloid plaque. And that's what happens in Alzheimer's disease. You have lots of these amyloid plaques. So amyloid precursor protein is one example of a protein that gets cleaved in this manner. There's another protein that's really important for sort of development, like developmental growth of neurons, named Notch. And Notch, similarly, the receptor for Notch can be cleaved by TACE and then by gamma secretase. So there's probably, I'll just off the top of my head, maybe 15 or 20 other proteins that have been recognized to undergo this sequential cleavage by these 2 enzymes. Yeah. Yes.
That raises the interesting question. You've got these enzymes that can cleave multiple receptors. How come they're not cleaving everything all the time?
How come the enzymes are not? Oh, well, yeah, very good. So the enzymes are thought to be regulated in a variety of ways. So there's, so for example, TACE has a region that's susceptible to what's called phosphorylation. So phosphate groups can be put on it. And so if a signaling cascade phosphorylates TACE in just the right way, that can turn it on or turn it off. There's actually been some studies showing that reactive oxygen species can stick to TACE and oxidize these cysteine residues on it, and that can turn it on. And so that's actually a hypothesis we have also about how this could be happening, is maybe it's getting directly oxidized and that's what's turning it on. But so the thought is that these enzymes in their natural state are sort of less active or inactive, and that it takes some type of signaling cascade to trigger, a conformational change in their active site that leads to them, performing their cleavage.
I guess what I was thinking of was once you activate it, you're going to be creating a variety of receptors, not just one kind.
Yes, that's correct. So as far as we know so far, it doesn't seem that there's only sort of a limited range of specificity. Yes, so these, tastes. So, when we blocked TACE in our experiment, in reality, we were probably blocking cleavage of Notch and APP, and some of these other proteins too. Yeah. Yes, Jen.
I know the neurobiology is very limited and ancient, but it's my understanding that initially there's like a lot of, like in early development, there's a lot of connections formed and then they get pruned back over time.
Yeah.
Are these mechanisms that you're talking about here kind of part of a normal healthy development, or are they only implicated in these disease models?
Yeah, that you're absolutely right. So actually, that was sort of how I first got interested in P75. So when I first started on my graduate work, I wasn't studying disease. I was trying to understand P75 indeed does cause certain neurons to die during just natural neurodevelopment. It kind of helps to prune away those extra neurons that you don't need. So I initially started with the goal of understanding that. But then what we realized is that a lot of these same pathways become reactivated in response to injury or in association with disorders. And I kind of started to gravitate towards that. And so my work kind of shifted. But indeed, cleavage of P75 has been shown to be important for causing death of several of neurons in several different regions of the nervous system as part of just natural neurodevelopment. Yes, so there is some similarity there.
Are there any other questions? Any questions in the chat? All right. So thank you very much, Dr. Kraemer. It was an awesome seminar. Thank you again.